Welcome to this Geneious Biologics video on clonotypes and how researchers use them to monitor clonal expansion and identify antibodies with high binding affinity. A clonotype is a group of B cells assumed to have originated from a single, naive B cell via clonal expansion. The antibody heavy chain sequences extracted from clonotypic cells generally share the same V and J genes, and are similar but not identical across the complementarity determining region 3, or CDR3 sequence. The CDR3 of the heavy chain roughly corresponds to the D gene segment. The CDR3 sequence is the antibody region that exhibits the most diversity and contributes the most to binding specificity. There is debate about what similarity thresholds on the CDR3 sequence determine a clonotype. Many immunologists use an 85% similarity threshold to account for somatic hypermutation. However, some researchers consider identical antibody sequences clonotypes, while others consider anything more than 70% similar to be clonotypic. After being exposed to antigen, the naive B cell experiences antigenic activation and clonal expansion, rapidly increasing. During this process, the multiplying cells undergo somatic hypermutation within the CDR3 sequence, causing the DNA sequence of the CDR3 to mutate at a rate 1,000,000 times greater than the normal background mutation rate. In contrast, the rest of the antibody sequence is largely maintained, hence clonotypes being defined by the same V and J genes, but a lower threshold of similarity across the CDR3 or D gene due to somatic hypermutation. This somatic hypermutation leads to variation in binding affinity within the proliferating B cells. Not all mutations result in improved binding. Cells that don't bind antigen tightly undergo apoptosis. B cells, whose antibodies bind more tightly to antigens, are therefore selected for during somatic hypermutation. This is referred to as affinity maturation. Researchers use clonotyping to monitor affinity maturation and clonal expansion after antigen exposure. This allows the researcher to identify antibodies with high binding affinity. By comparing clonotypic antibody diversity, researchers can gain insights into the similarities and differences within and between individuals. For example, researchers can characterize the frequency of clonotypes in an immunized subject and identify which naturally occurring antibody sequence has the highest binding affinity. Clonotypes that occur at higher frequencies are likely selected for during B cell maturation. Researchers can also use the clonotype characterization of 2 immunized patients to study the similarities and differences in the adaptive immune response between unrelated individuals. If you're interested in analyzing and identifying clonotypes in your antibody sequence dataset, Geneious Biologics has a suite of tools to take your data from raw sequence files to annotated and analyzed results and graphs. For more information about Geneious Biologics, visit geneious.com/biologics.